Thank you very much for the warm introduction and welcome. It's a real pleasure to speak to you today about some of the work that's been going on in New Zealand. Although the title that's in your books there talks about me talking about a New Zealand and international response, I'm really going to focus on New Zealand today, telling you some of the work that we have going on here. And I'm going to share several different people's work very, very briefly in the 20 or so minutes, and then I'll hand over to Tim and we'll have a Q&A at the end. These are three of my heroes, and great, Baz, always difficult actually to follow such a great speaker as Baz, but these are three of my heroes. We know about Parkinson's disease, or at least the first major treatment for Parkinson's disease, because of these three people. And the person there, Arvid Carlsson, was the person who discovered dopamine. In 1957, he discovered something which looked awfully like dopamine as we know it today. Lots of people weren't overly certain that he'd in fact discovered something unique from what was already known. And so 2 years later, 1959, he published almost an identical paper in the same journal, convincingly showing that he'd discovered dopamine. And then just 2 years later, Ole Hornekiewicz, who worked in Austria, identified that it was dopamine that was missing in the brains of people that had died with Parkinson's disease. And then within about 1 year, the person on the bottom right for you, Oliver Sacks, who many of you would probably have read his books, The Man Who Mistook His Wife for a Hat and Musicophilia, and other books that have inspired neuroscientists for generations. He first started treating people with levodopa, in fact with dopamine, because he thought, well, if that's what's missing, then that's what we should treat people with. And that was 1962. Now, the reason I have him in a pair of Speedos there is because he was invited by Vanity Fair, the well-known magazine, to talk with a reporter, and he said, if you want to have a talk with me outside of work, I'll meet you where I go swimming, and we'll have an interview there. And they snapped him in his trunks, and this became the picture on Vanity Fair. An incredible person. So actually, when it comes to looking at some of the wins in Parkinson's disease, just 3 years from the discovery of dopamine to having something that helped people live their lives with Parkinson's disease is actually remarkable, isn't it? Isn't that incredible? But of course, that was 60 years ago, and so we'd like to see how quickly we can move things on from there. I just want to show you quickly, because you'll hear these words a lot and you've probably read a lot about these things. Lewy bodies are the accumulations of a protein called alpha-synuclein that accumulate in the brain, in the neurons and around the neurons in the brain in Parkinson's disease. Now, to give you an idea, Lewy bodies were identified first in the early 1900s. However, alpha-synuclein was discovered in 1997. I remember reading the paper early on in my career that the contents of those Lewy bodies had actually been discovered. So it's quite recent. I hope the people that discovered it get a Nobel Prize for it. I think it was an incredible discovery. And so that's what we think of as the pathological basis for Parkinson's disease. Now, this is a slide that was actually largely put together by Dr. Daniel McGowan, who's with us today, and then I butchered it a wee bit to look a little bit different. But what this shows is that there are many different ways that we can think about the idea of a treatment or a cure for Parkinson's disease. We can think about the left side here, which is things that we can do that change the way that our brain works pre-PD, right through to having other things that can be applied, or at least experimentally, and some of these are clinically available, in late-stage Parkinson's disease. And I want to show you today that New Zealand is actually working in areas across the spectrum. Yesterday we had some great talks with Baz, and a lot of the scientific community got together. And actually throughout the day we saw that there are projects going on in each of these areas that we think will make a difference for Parkinson's disease, short-term and long-term. Now, I want to just highlight something that Rich mentioned earlier, and the Neurological Foundation have supported the Human Brain Bank for 30 years. And this has actually been really influential in New Zealand for the development of the neuroscience, one aspect of the neuroscience program that goes on in New Zealand. And this has been supported by incredibly generous families who decide to donate the brain of a loved one when they pass away, and has led to many discoveries that have happened in New Zealand. And I'll just point out that Sir Richard Fall is here. He developed the brain bank, and I've had the privilege of coming on board subsequently to work in this incredible facility. What I want to tell you about is something that Buzz has already touched on, and this is the area of my research. And that is the idea that in Parkinson's disease, the disease probably or possibly begins in the olfactory system, which is area 1 there, followed by it moving to area 2, which is also part of the olfactory system, 3, which is part of the olfactory system, and then 4, which is the substantia nigra, which takes someone to the doctor with a movement disorder, and then eventually 5 becoming affected, which may produce a cognitive disorder in Parkinson's disease. The problem with this is that we know almost nothing about the human olfactory system. Even though we've been poking cotton wool buds to try and detect COVID up our nose for the last few years, billions of times as a nation, we don't actually know too much about the territory that we're poking those things up through. And so my research has been focused on understanding the early origins of Parkinson's disease in the olfactory system. Now, this is the picture that you'll see if you look up Wikipedia about the olfactory system. And in the background there, you can see a human brain, a human head, and in the foreground, it looks like all of the information that's contained there is actually from the human, but actually 100% of that data is from the mouse. So Wikipedia is even telling you that you're a big mouse. Don't believe it. We've got a lot of work to do, but actually humans use their sense of olfaction in much the same way as an animal does, allowing the scent in one nostril to strengthen, and so you go towards something, and then when it weakens there and strengthens on the other side, it comes back, you come back. And graduate students at Berkeley use exactly the same technique for following traces of chocolate on the ground. And you can see at the bottom there that actually the more days they do it, the better they get at it. So surely chocolate will be a treatment for Parkinson's? No, maybe not, I'm not sure. Okay, so what I want to show you now is that alpha-synuclein, that problematic protein in Parkinson's disease, shows up in the olfactory bulb. And the red stuff here, the blue is the olfactory bulb. The red stuff at the top there is alpha-synuclein, accumulating in abundance in the olfactory system very early on. Now, I want to show you something else that I think is amazing. And that is that if you look at the olfactory bulb, and here's just a small slice through it, what we can see is this label called tyrosine hydroxylase, which basically means dopamine, not quite, but close enough, means dopamine. There is a lot of dopamine in the olfactory bulb in Parkinson's disease. So this says that maybe the dopamine factor in Parkinson's is not that it's inherently a disease of dopamine, it's just a disease of susceptible cells that happen to produce dopamine. So in Parkinson's disease, even in very late stage, dopamine is not affected in the olfactory system. So one of the things that we've been doing in this area has been to try and reconstruct to understand what the olfactory system looks like in the human brain. And here we have a slice showing both olfactory bulbs, as if you'd taken a slice down through the brain like so, and the areas in the middle here, and sorry on the sides it's going to be a bit hard to see, this is where the air goes through into your lungs eventually. But these brown bits here are detecting the sense of smell as we breathe things in and breathe out. And so by taking several thousand sections through the human brain, or this part of the human brain, and then reconstructing them using artificial intelligence, and then teaching computers, the biggest computer in New Zealand, how to pull the interesting information out that we want to, we have been able to develop a way of visualizing and understanding what or at least to a fair degree, understanding what the olfactory system looks like in humans. This is 1,234 sections through the human olfactory mucosa, which eventually can be turned into a model which we can then compare between normal and people that don't have Parkinson's disease and people that do have Parkinson's disease. What is going on very early on in this region in people with Parkinson's disease? Now, the ultimate goal here is that if we could reduce the burden of alpha-synuclein in this region of the brain very early on, when that burden is out here in the periphery rather than in here in the central part of the brain, in the future I imagine a world where people go to the doctor with a smell disorder so that they never have to go to the doctor with a movement disorder. Wouldn't that be incredible? That would be amazing. So that's the work that we are trying to achieve. And these models have already been put to good use in ear, nose and throat clinics because we haven't had these anatomical maps before of the upper airway here. Now, I just want to show you also that we've been able to trace, and it's a pretty thin yellow line there, but you can see the yellow line on these graphs showing just where within the olfactory system alpha-synuclein turns up. And what we can say is that it specifically targets the olfactory sensory neurons. We think that the smelling neurons are the ones that preferentially pick up alpha-synuclein early on. Now, that's the very early phase of this schema that I've got here, looking for very early detection. The question is, how do we clear some of that alpha-synuclein from the middle section here, where we might want to get rid of the alpha-synuclein so that it doesn't get back to our substantia nigra and doesn't affect our thinking and our movement and so on. Well, I'm just going to spend a couple of minutes showing you some of the work that we're doing here, and I want to introduce the concept of the nose being a window to the brain. There's some things that we're doing. We are culturing cells from people that are newly diagnosed with Parkinson's disease and their live-in partner, because my fun fact of the day is that if you change your microbiome, others that live with you will change their microbiome. If you take antibiotics, your dog, if that's in your animal, will change, if that's in your house, will change its microbiome. If the dog takes an antibiotic, your microbiome will change. Who we live with makes a difference to our microbiome. And so comparing someone diagnosed with Parkinson's disease and the person they live with, taking those cells and growing them so that we can then have a model of Parkinson's disease in a dish has been really important and something that we're actively doing. But what we are specifically trialling here, and this is work actually done by a woman, Dr. Karen Ichikawa, has been to develop these drugs called proteolysis targeting chimeras. And what they are is essentially a drug where one side of the drug binds to alpha-synuclein and the other side of the drug binds to the part of the cell that will disintegrate alpha-synuclein, chop it up. And then the side that will help chop it up drags the alpha-synuclein towards the chop-up part of the cell, the recycling part of the cell, so that it can, rather than accumulate, be chopped up into small bits and be recycled by the cell. So this is an incredible technology that several groups worldwide are using, which I think will have great impact in the future. I would just show you a couple of exemplars here of Protax, and then just to show you here an example of this working to a degree. It's not there yet, but here you can see the high levels of alpha-synuclein, the bright green in cells normally, and when you expose these cells to high levels of Protax, you reduce the level of alpha-synuclein. So this is something that is doing what the scientists who developed it want it to, but it's not quite there yet, but a work in progress. I'd like to now just go over to more of the right-hand side of the curve here and look at the antipsychotic aspect here. One of the challenges that you will be aware of as people who have Parkinson's disease is that treating the hallucinations that sometimes come with Parkinson's disease or the psychosis that sometimes comes with Parkinson's disease is really difficult. And the reason for that is that most antipsychotics dampen down the effect of dopamine. And you can't do that if you've got a dopamine deficiency. You need to maintain the dopamine levels while also bringing the hallucinations down. And so there is that was not where I thought it would go. I'm going to come back to that one in just a second. There is a drug that came out recently that for the first time preferentially targets a different system, serotonin, and dampens down the serotonin system so that people with Parkinson's can maintain their dopamine system. The problem is, and you can see here from the top number there, this is a drug called Nuplazid, is the number of serious side effects and even deaths from taking this drug, which is not approved in New Zealand, is actually incredibly high compared to the much safer other drugs that people with Parkinson's disease might wish to take, or might be prescribed, I should say, and so this is a real problem. So some of the work that has been going on in the Sperry lab, Jonathan Sperry is a medicinal chemist, has been to identify other drugs that are responsible for dampening down the serotonin system without affecting the dopamine system. And I'll just give you an example here of one of these drugs that he's been working on. This is one that, through a detailed international screen done independently, has been shown to at very, very low levels affect the serotonin system, and 5-HT2A there is the serotonin system. At very, very low levels, it will affect that system, which is great. And then only at much higher concentrations do we first start to see the dopamine system being affected by this particular drug. So you can give that low level and affect the serotonin system and leave the dopamine system unaffected. This will have, I hope, in the future, it would be great to see that this has significant impacts on people that have those symptoms in the future. Now, if I could trouble you just to click back a couple to the one that I confused, back another one more. Thank you. Here is work that we heard about yesterday, and I asked if I could share this today. And I'm not going to go through this slide in great detail, but I do just want to point out that it's been identified in this paper at the bottom here by John Reynolds and his team that actually the timing of receiving dopamine is really precise. So having dopamine there constantly can be problematic. And we see that in Parkinson's disease, if you take dopamine or levodopa for a long time, you can end up with all sorts of side effects and so on. And some of that is, none of those side effects are helpful. And so what he has been working on is the idea that if you could, in fact, get dopamine just to act in this 1 to 2 second area, you could in fact potentially avoid some of those side effects by only allowing dopamine to work in a temporal manner, in a time-dependent manner. And so he and his team are working on a system for encapsulating dopamine into ultrasound susceptible packages, which you then take. And with ultrasound, you can release the dopamine in a temporal manner, much like the body would be doing in a minute-by-minute kind of basis, or in fact second-by-second basis under normal physiological conditions. So I think this kind of technology, ultrasound susceptible capsules for dopamine, may be important in the future for allowing people to have good effects of dopamine without all of the side effects that come with taking something like levodopa. So I'll skip on, in fact I can skip on from there. So I just really wanted to point out that there are a couple of areas there that we are working on in New Zealand, not me, but others in New Zealand are working on very early on in Parkinson's disease, in the middle phases of Parkinson's disease and towards the end of Parkinson's disease, that I think gives us good reason to hope. There's hard work yet to come, so hope and hard work, and then a little bit of luck, and I think we will see differences in the next few years in the way that Parkinson's disease might be treated. So I'd like to say thank you to some amazing people. That beautiful fly-through of the olfactory system. The star of that show was Victoria Lowe, an incredible scientist who works in my group. I've shown you a little bit of the work of Dr. Karen Ichikawa, also of Jonathan Sperry, and also John Reynolds. They all have their own groups. And there's so much more work we could actually talk about of what's going on in New Zealand in the Parkinson's disease research and treatment space. And then, of course, there's the international work, and I know you'll hear some more about that over the next few days. Just a special thank you to all of the people who have funded the work that I've presented today. Cure Parkinson's Disease, Cure Parkinson's New Zealand have been major sponsors of the work that I presented. Thanks very much, and I'll hand over to Tim.
Thanks, Maurice. Look, it's pretty warm in here and you've been sitting for a while, so I don't mind if you want to stand up for a minute and I can sort of chat a bit and then we'll sort of hook into it. Just while you're doing that, I just wanted to mention it's been a privilege to have Bas here, Professor Bloom. He came to Christchurch and very graciously sat in on some of our PhD students' presentations. Some of them are nearing the end of their thesis, and really gave some great feedback and advice and even encouraged some collaboration. So I think for those who helped bring Bas out here, it's going to have ramifications well beyond this talk that he's just given. So I'm also going to give a New Zealand response, as Maurice has done, and perhaps talk mainly about the work we do in Christchurch. So, the theme was the pandemic of Bas's talk, and so I thought I'd give a New Zealand perspective about the epidemiology here as far as we know it. Two of our senior researchers a little time ago used the national prescribing data that they could obtain from Wellington about the national prescribing habits of drugs that you use for your Parkinson's, to work out what the incidence and prevalence of Parkinson's is in New Zealand. And Bas took us through the bath with the water coming in and out. Using that data, they figured out that the peak incidence, in other words, what's the peak age that people will present in New Zealand with their Parkinson's, is about 80. When I was a registrar, it used to be thought to be about 59 or 60. So we know that people, the peak age of incidence of Parkinson's in New Zealand, as far as we're aware, is 80, and the prevalence, in other words, the number of people who have Parkinson's at any one time in New Zealand, including folks here, is 85 as the peak prevalence. You'll be interested to see that actually there's a tail at the end that goes down, and I noticed on Bas's slides he also had a tail that goes down in the oldest old. And it looks like, once you've weathered your 80s and you ain't got Parkinson's, you may be, more spared from getting Parkinson's. You might get other things, but less likely perhaps to develop Parkinson's in our oldest old. And then what Tony and Daniel did was they looked at ethnicity. Now remember, this is in the prescribing, so it's not, we didn't go out door to door knocking, so we've got to take this with a wee bit of grain of salt because it all depends on access to resources and so forth. But if you look at the lines there, this is the prevalence at any one time, depending on your ethnicity, as to how many people or proportion of people in the population who have Parkinson's. And in essence, the top line is European New Zealanders. The very bottom purple line is our Maori population, and in between are Asia and Pacifica. And on the face of it, it looks like our Maori population has half the incidence, half the prevalence, I should say, of Parkinson's compared to European whites. Still need to sort of tease it out a bit, but that may be real, or it may just be a factor, as I say, of access to resources and not getting prescribed medications. But it's a very interesting observation that really needs follow-up. When they looked at the regions, the DHB region that is, they still exist, the DHBs, they looked at the prevalence by region, and you all have your own regions, but most of you, I guess, are up this way. and the highest prevalence seemed to be in South Canterbury and Canterbury, at least when we look at the drug prescription data, and the lowest, as you can see there, in counties Manukau. You might think that those top ones there are perhaps more rurally, a greater rural population in those particular DHB areas, but we'll come back to that, but that may be an explanation. When we look at the incidence over time, and this is, the data you saw is a bit older, now this is looking over time, from 2006 to 2017, and in essence you're looking at basically the incidence over time, males and females broken up, and the top one is the overall population. Actually, as Bas already told us, the incidence is not really changing much over time. So there must be another reason for the higher prevalence, which is going up. So the incidence, in fact, if anything, that's going down a little, the incidence. That's the number of people per year developing Parkinson's. And we've projected forwards from the latest data, and so we figure, someone mentioned 12,000, it's probably 13,000 people with Parkinson's at the moment, or that was back in 2018. And we figured that back in 2017 the numbers are expected to double by 2014, looking at the trajectories, which is really what Bas has been saying, and that this is a pandemic. Now, Parkinson's, as you all well know, is a multi-system disorder with lots and lots of problems associated, motor, non-motor, autonomic, pain, and so forth. And we can't escape from the fact that a proportion of people with Parkinson's do get dementia as time goes on. And I think we have to confront that, because this is a problem that we really need to try and fix. We need to fix Parkinson's, but in particular, in my view, we need to try and prevent people getting dementia with their Parkinson's. And there's been papers talking about the inevitability of dementia if people live long enough. Well, without going through all that slide and the reasons how we could calculate it, but we looked at the New Zealand scene, and we estimated that in New Zealand the lifetime risk of someone with Parkinson's developing dementia is about 50%. So you mustn't think that everyone's going to get dementia, but you mustn't think that you're never going to get dementia. There is a chance. And now, why our figures are lower than others published elsewhere in the world, and we're not sure why. Maybe we're living a healthier lifestyle here, maybe we're dying of something else, and before we've had a chance to get our dementia. Just like men, if all of us live long enough, we're all going to get prostate cancer probably. But anyway, that's a better figure than internationally, so I think there's hope there. Actually, if you looked at that part of that graph, you would have seen that the age of death, the mean age of death of people with Parkinson's, is very similar, almost identical to the average age of death of people without Parkinson's. Very similar. Curious, isn't it? Which is a little bit against some of the international figures, but it's in the 80s, as you can see, some mid-80s. So here in New Zealand, it seems that the mean age of death is pretty similar between Parkinson's and non-Parkinson's. I'm now switching to a study that I'm hoping that some of you are part of, but I'm hoping that many of you with Parkinson's here are not part of, because it gives you a chance to do that. So this is about a study we're partway through, and so I'm telling you about some results which are really hoping not to bias the people who are going to join the study, but I thought it'd be interesting for you to know, and perhaps stimulate some of you who aren't part of the study to join, because it's a national study that we're doing based in Christchurch, but national, and we're trying to look at the New Zealand scene in terms of the environmental influences, including pesticides and herbicides and who knows what, and the genetic contribution. I mean, in essence, we're partly a genetic disorder, but clearly, and we've published a paper a year or two ago on movement disorders, there are a number of hits that people have over their lifetime that add up to getting Parkinson's in someone who's already a bit susceptible for whatever reason. And so this study is a nationwide study. We're hoping to recruit 500 people with Parkinson's from around the country and 500 people off the electoral roll who don't have Parkinson's, and try and tease out what are some of the lifestyle factors, some of the environmental exposures, and some of the genetic factors in our New Zealand scene. And let's face it, every country is a little different. It may lead to people getting Parkinson's here in New Zealand. There's a number of people involved, and those are the folks there. I want to thank all the neurologists around the country and the Parkinson's New Zealand and the various societies up and down the country. who are helping us. So as mentioned, we're trying to recruit 500 people with Parkinson's. We've got about 300 so far. And those of you who've been enrolled, you'll be aware of what it involves, which is quite a lot. It's all done, there's no visits as such, it's all done online. And you get sent a study pack, which allows you to test your sense of smell. That's the upset. You produce some saliva and you send it back to us, and we send it off to the States to look at all the genes, a huge packet of genes that we know can contribute to people getting Parkinson's. You fulfill quite a number of questionnaires, and especially your exposures over your lifetime, potentially to a rural environment, to potentially pesticides and the like. So how many people here actually are part of that study? Oh, fantastic. Yeah, it's a great thing. So that's a lot more than I had thought, and thank you for doing that. And it gives you scope for the people who haven't put up their hands, who do have Parkinson's, to join in, and you'll see the website there. That's how you can enrol. It's all done at home, but it takes a bit of time, and we'd be grateful if you would look at that.
What are your criteria for the study?
The questions, the criteria for the study, having Parkinson's. And actually, no other criterion. Having Parkinson's, it's got to be diagnosed, and even people who've got what we call atypical Parkinson's, MSA, PSP and corticobasal syndrome, but predominantly Parkinson's. No specific criteria, you just have to be able to get online, one way or the other. Anyway, so far we're about 2/3 through our collection. Thank you for those who have joined and those who are going to join from here. And a similar number of controls. As Bas has already told us, it's more of a male condition than females, so 2/3 of our contributors are male. And we're not going to go through the regional contributions, but there's a fair swag from Auckland, thank you. Yeah. Just on the right, really, you just see the average age of people is about 70, who have had your Parkinson's for about 6 years, with an age at diagnosis of about 60, which is a bit younger, as I told you about the prevalence. So, the really older folks who might struggle more with the sort of internet and so forth are perhaps not joining as much as the more vigorous people of my age. Just a few things, and really just to back up, and Bas has already mentioned this, but it's good to know about the New Zealand scene. We looked at, as our interim analysis of some of the results, smoking, coffee, alcohol and head injury. We've had all those mentioned to us already by Bas. And without going through the graphs, in essence, we found that people with Parkinson's were underrepresented in smoking. Right? So we've already heard that smoking has some protective effect against getting Parkinson's. And that's the case in New Zealand as well. I should mention, and Bas mentioned, you shouldn't start smoking. In fact, we know that people who smoke, once they've already got their Parkinson's, do less well in the future, particularly with their cognition. So, you may have been protected by smoking before you got your Parkinson's, but if you are smoking, you should stop. What about caffeine? The same applies for caffeine. So people who don't drink coffee have a greater risk, it seems, to develop Parkinson's, and that's the international literature. Alcohol, we found that there was a lower alcohol consumption of people with Parkinson's. In other words, this is in their lifetime, not people once they've got Parkinson's. So in our figures anyway, somehow there's a lower alcohol consumption in people who get Parkinson's. This sort of smoking and alcohol and caffeine reminds me of George Best. I don't know if anyone remembers George Best was a fantastic soccer player, probably the best in the world at his time, amazing. But he liked to drink, and he made a famous statement once, which the line, something like, I involved in sex, drugs and rock and roll, and the rest of the money I wasted. I spent most of my money on sex, drugs and rock and roll, the rest I wasted. And then he got liver failure, actually. Yeah. Anyway, these are the figures that we have so far, and head injury also, as Bas mentioned, there seems to be a higher rate of head injury in people who have developed Parkinson's, at least from everyone's responses, than people who've never had a head injury. So it does look like head injury, as internationally, also as a risk factor in New Zealand. So these are fairly reflective of international studies, but it's good to know that seems to be happening in New Zealand. But we still need to collect more data. The genetics, and Bas touched on the genetics. I'm not going to go through the genetics. He mentioned the GBA gene mutation. which would be the commonest mutation in New Zealand that would be associated with Parkinson's. It doesn't necessarily cause Parkinson's in itself. In fact, it usually doesn't, but it gives you a higher risk of developing Parkinson's. In our own Christchurch cohort, which we looked at closely, there's about 10% of people with Parkinson's have this gene mutation. There's about 100 or more mutations of the GBA gene. Some are more, if you like, pathogenic. They're more likely to cause Parkinson's or contribute to Parkinson's than other mutations. So, it's not a binary thing. There's a whole lot of different mutations. But anyway, 10% of Christchurch cohort that we've looked at closely have a GBA mutation. So far in this particular nationwide study, about 5% have recorded, or we found a mutation in the saliva of 5% of our respondents who have Parkinson's, and only a tiny proportion of people who are controlled. So we think that figure might rise as time goes on. Now, contrary to what we might have expected, so far, these are interim results, no association with rural living over the years, no association with well water, which has in other studies internationally been associated with a risk of Parkinson's, probably because of the pesticides and herbicides and so forth. And actually, at the moment so far, Bas, and I'm sort of embarrassed to say this almost, that there's so far no obvious association with pesticide exposure, but this is only a yes-no answer. So, within the pesticides, there's a huge range. There's going to be different pesticides, as we've heard, that are going to be far more pathogenic, if you like, than others. And we'll just have to wait and see. So this is not a reason to just relax and think, oh, well, pesticides. I think I'd be more persuaded by the international literature and by Bas's own presentation that, let's avoid pesticides. Surely there is something there. But at the moment, we haven't been able to show that in our questionnaires. Remember, we haven't gone out and tested this in people. Maybe we should be. Maybe there's a hair or sample or something that one could do. I don't know how you test for pesticides, but Bas might be able to tell us. So caveat, these are all provisional results, unpublished, but really just to let you know what's happening, and really great if people could join. Okay, I'm switching tacks briefly. Andrew asked me to mention something about the drugs that we use here in New Zealand, and that's another 2 or 3 lectures in itself. As you're aware, lots of drugs, lots of side effects, and I'm just going to mention one that many will be aware of, but not necessarily all. Incidentally, this is just a list of the potency of the medications we use for Parkinson's in terms of improving motor function. So we know that levodopa is more potent in improving at least motor function than the dopamine agonists, pramipexole, ropinirole, and more so than rasagiline. So if you're going to choose a drug that you want to, is the most powerful, if you like, then levodopa is the drug, and we know that, but and has fewer side effects than the dopamine agonists, but the two work together well. I just want to mention one of the side effects that hopefully some of you, and perhaps hopefully most of you who are taking drugs are aware of, and that's something we've become more and more aware of as prescribers over the last decade or so, about impulse control disorders. These are particularly related to the dopamine agonists, pramipexole and ropinirole, and worldwide, about 13% of people have significant impulse control disorders from their drugs that they're taking for their Parkinson's, and particularly the dopamine agonists. And particularly, and especially in males, pathological gambling. In other words, gambling away, and I've had patients myself who've gambled away the family fortune, and it's disrupted marriages and relationships. Hypersexuality, I've had people who, internet porn, prostitutes and so forth, who completely out of character. And then people buying things they don't need, 3 cars, we only need 1, 50 pairs of shoes, sort of Milton Marcus type behaviour. As a compulsive shopping. So these are potential problems that are not due to you or the person with their personality. It's effect of the drugs driving that behaviour, and you need to be aware of it as a partner as well as a patient, because it can creep up on you, and the person who's doing this has little insight, and they're just driven to do this, and it's very hard to change the behaviour. You need to be aware of it as a partner. And if so, we have to take the person taking that drug off that dopamine agonist, basically. Otherwise, it'll continue. Punting is a different thing. That's come from the crack literature, drug addicts. But in Parkinson's, people who like to take too much dopamine, and some people do, they just hate being off, they hate switching off, and so they take more and more dopamine, levodopa, throughout the 24-hour period of the day. Generally get very dyskinetic as a result and hate being off, and then they just overuse the levodopa and start to do sort of stereotypic behaviours, in other words, sort of habits over and over again that have no earthly use, but they feel compelled to do it. So especially I have male patients who will take the motor mower apart, put it back together, take it apart, put it back together, for no earthly purpose, but feels driven to do it. Other perhaps women, but this is not, not going to be stereotyped here, may have more domestic things like tidying up and tidying up. These are repetitive behaviours that have no earthly purpose, often 24 hours a day. Some people are compelled to look at the internet to no earthly purpose. Those people are taking too much levodopa and need to cut down the levodopa. Okay, that's the end of that lesson. I'll just mention the drug, while we're on the drugs, the trends in drug use in New Zealand briefly. Some of this is old data, but I'm just going to draw your attention to some of these lines. This is Tony and Daniel again, looking at the trends of prescribing over the years. And some of you would have been on these medications probably from the 1990s. And you can see over time on the bottom axis and the particular drug type on the top, some things are getting more prescribed, some things are less. Levodopa, as that middle line, is increasingly prescribed in New Zealand, as you might expect, with the increasing prevalence. Some drugs are going down, like the anticholinergics, but let's just move on to individual drugs. So this is an interesting one. Total levodopa is going up, but there's a difference between the two brands that we have available to us, between Sinemet and Matapar. I've got no idea why there's a preference in prescribers, this is obviously prescribers, for using Sinemet over Matapar. They're both equally good, so I don't think you should be switching because more people are on it, but that's just a curious observation. The anticholinergics, which we used to use a lot for tremor, have got a bit of a bad press because they can impair people's memory, not permanently, and are now being prescribed less and less and less. Incidentally, these graphs on the right are the more recent data, but we don't have the figures like we have on the left. But you can see that the trends are continuing. So amantadine, Barry Snow here was one of the first to show that amantadine was good for dyskinesia, and that's getting increasingly prescribed and still so right up to the present day. It's a good drug for dyskinesia, can cause issues with memory and hallucinations sometimes. The dopamine agonists, ropinirole, pramipexole, increasing, especially pramipexole, start to get used later. Some of the old ones have died off. And apomorphine, which some of you here might be on, it's a short-acting dopamine agonist that gets pumped in under the skin on a continuous basis. That's getting more and more used, which is great because it is a very good drug for people who are having trouble with a lot of fluctuations. And finally, this is important, and I think that's really good this is happening. The drugs for hallucination psychosis, we've already heard from Maurice about that already, are being increasingly prescribed in Epizil, and now the patch rivastigmine are being used increasingly for hallucination psychosis, which is really important because these things can be really debilitating, as somebody who has experienced these things will know. And also equally important and really good to see is that the other drugs, I've got them the wrong way around actually, I have to tell you. The rivastigmine and epizole are for memory and confusion, and the other ones, clozapine and quetiapine, are for hallucinations, so somehow I've shifted them to the wrong spot. Anyway, both these classes of drugs, which are really important in people who are struggling with memory, confusion, getting into dementia, some people, or hallucinations, are getting increasingly used. So it's awareness by the physicians around the country that we need to use these medications to help people with these really troublesome neuropsychiatric side effects. So I'm not going to talk about our own research because I've run out of time, but we do a lot of research in Christchurch, but it's related, as Rich said, biomarkers of cognitive impairment especially. We bring people in on a longitudinal basis every 2 years, and they're very generous with their time, and we assess them to the hilt with neuropsychological tests, scans of various sorts, and hoping to find real predictors of how people are going to go over the years, with the hope that we can, a person who turns up with their Parkinson's initially, we can say or project how their course might be based on the whole of biomarkers, which will be important if we've got good drugs to slow different aspects of Parkinson's down. So you may find a new medication that's going to slow down one aspect of Parkinson's but not the other. So it's going to be, I think, as Bas would have mentioned previously, a cocktail probably of medications that are going to help us try and treat these wretched symptoms that people will develop in different ways in different people. So that's a brief fly-through, and I've gone over time, whereas Maurice finished exactly on time. So I'll call it quits there, and I think there may be time for questions. I'm not sure. Anyway, thank you for listening.